Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas. In today's class we are going to discuss the head and neck development. Learning outcome for today's class are that the sources of the mesenchyme forming the head region, then the development of the pharyngeal apparatus and their congenital anomalies. So what are the sources which are forming the mesenchyme of the head region? These are usually four sources: the paraxial mesoderm, the lateral plate mesoderm, the neural crest cells, and the ectodermal placodes. So what the paraxial mesoderm is forming, it is forming the membranous and cartilaginous components of the neurocranium. Here in this diagram of the skull, you can see this area which is shown in the light green color area. This is the area forming the neurocranium, and this area which is shown in dark green color, this is the viscerocranium. So this neurocranium is formed, its membranous and cartilaginous components are formed by the pharyngeal mesoderm. Also, all the voluntary muscles of the craniofacial origin are formed by the pharyngeal mesoderm, the dermis, the connective tissue in the dorsal regions, the meninges, caudal to prosencephalon. So these are the derivatives which are formed by the pharyngeal mesoderm. Next, you can see that the lateral plate mesoderm, what it is forming here, you can see these are the, in yellow color you can see the laryngeal cartilages. So this one is the arytenoid and the cricoid cartilages which are formed by the lateral plate mesoderm, and also the connective tissue in this area, so it is formed by the lateral plate mesoderm. So here you can see this hole in red color is the neurocranium, this hole in blue color is the viscerocranium, and in yellow color you can see these are the laryngeal cartilages, the both arytenoid and cricoid, which are formed by the lateral plate mesoderm. Then the third source are the neural crest cells. This is very important. It originates in a neuroectoderm of the forebrain, midbrain and hindbrain regions, and they are forming the entire viscerocranium. This entire viscerocranium is formed by the neural crest cells. Its membranous components, its cartilaginous components, they are formed by the neurocranium from the neural crest cells, but also some of the parts of the membranous and the cartilaginous components of the neurocranium is also formed by the neural crest cells. So here you can see this one is the neurocranium which is formed by the pharyngeal mesoderm, and this part of the neurocranium is formed by the neural crest cell, and this whole area is the viscerocranium which is formed by the neural crest cell, and these neural crest cells also contributes to the pharyngeal arch skeleton. So the neural crest cells move from the area of the developing brain in a form of the population. The first population migrate, and it has two fate. Either it has the neuronal fate or it has the non-neuronal fate. So the cells which have the neuronal fate, they will form the trigeminal ganglion, and the cells which are forming the non-neuronal fate, they will form the frontomaxillary mesenchyme, the mesenchyme of the frontonasal mesenchyme of the maxillary region and the mandibular region. Then the second population of the neural crest cells, which are now forming the otic ganglion and the mesenchyme of the second pharyngeal arch. Then the third population forming the ganglia of cranial nerve 9 and cranial nerve 10, and the non-neuronal contributions to the third, fourth and the sixth arches, and some even going to the outflow region of the heart. So this is the third population. Then the fourth population forming the occipital myotome. Here you can see the occipital myotome derived from the mesenchyme form the hypoglossal cords which will form their tongue. So fourth population from occipital myotome derived mesenchyme, they will enter into the area of the developing tongue. Now the cells from the ectodermal precodes, this one is the fourth source. So along with the neural crest cells, they will form the sensory ganglia of the 5th, 7th, 9th and 10th cranial nerves. So to revise, there are the four sources. The first was the lateral plate mesoderm, which was forming the neurocranium, its bone, the membranous components, the cartilaginous component. Then was the lateral plate mesoderm, which was forming the two cartilages, the arytenoid and the cricoid cartilages in the area of the larynx and its connective tissue. Then was the neural crest cells, which are forming all of the viscerocranium and part of the neurocranium as well. And the fourth source, which is the ectodermal placodes, they are forming the sensory ganglia of the 5th, 7th, 9th and 10th cranial nerves along with the neural crest cells. So these are the sources which are forming the different structures in the head and neck region. Now we will move on to the pharyngeal apparatus. The head and neck region of the fourth week human embryo somewhat resembles these regions of the fish of the embryo of comparable stage. This explains the former use of the designation the brachial apparatus. Brachial is derived from the Greek word the brachia or the gill. So what is the pharyngeal apparatus? This pharyngeal apparatus consists of the four components. These are the pharyngeal arches, the pharyngeal pouches, the pharyngeal grooves and the pharyngeal membranes. So look at this diagram. This is the embryo from the fourth week of development. Here you can see the development of pharyngeal arches over here. The first, second, third and fourth pharyngeal arch is present, and this is the area of the stomodium which will form part of the oral cavity. This one is the lens placode and the nasal placode, and here you can see the area of the developing heart. So these arches when developed in the floor and the lateral wall of the pharynx and consist of the thick core of the mesenchyme, which is covered by the ectoderm. So first pair of the pharyngeal arch primordium of the jaw appear as surface elevation lateral to developing pharynx. So this is the first pair which appear lateral to the developing pharynx. Soon other arches appear as obliquely disposed rounded regions on each side of the future head and neck region. So you can see this one is the first pair. These are the U-shaped mass which meet in the midline. This one is first pair, the second, third and the fourth pharyngeal arches you can see in this diagram. So what are the pharyngeal arch components? Each pharyngeal arch consists of core of mesenchyme. Here you can see in pink color these all are the pharyngeal arches. The first pharyngeal arch, the second pharyngeal arch, the third pharyngeal arch and the fourth pharyngeal arch. These consist of core of the mesenchyme, and this mesenchyme is covered on the external surface by the ectoderm, and on internal surface you can see the endoderm of the pharynx which is present. So in between the two arches on outside there is the cleft which is called the pharyngeal cleft, and on inner aspect you can see the outpouching of the pharynx between the two arches. This is called the pharyngeal pouch. So on inner aspect you can see pharyngeal pouch, on outer aspect you can see the pharyngeal arch, and this whole on outer aspect you can see the cleft which is present, and this hole is the pharyngeal arch. So this pharyngeal cleft is lined by the ectoderm, this pharyngeal pouch is lined by the endoderm, and core is formed by the mesoderm. So each pharyngeal arch consists of core of mesenchyme. It is covered externally by ectoderm and internally by the endoderm. In the third week, the original mesenchyme is derived from mesoderm. During fourth week, most of mesenchyme is derived from the neural crest cells that migrate into the area of the pharyngeal arches. As I have told you in previous slides, the neural crest cells migrate from the area of the developing brain and they form the core of the pharyngeal arches. Now you can see in this diagram that here at this stage only the first pharyngeal arch is present and second is in process of development. Then you can see the first, second and third are now developing, and here you can see the first, second, third and fourth pharyngeal arches are present. and in between these arches you can see the pharyngeal grooves or pharyngeal clefts which are present over here. So here you can see the pharyngeal arches which are present, and on outer aspect you can see between two arches there is pharyngeal cleft or pharyngeal groove which is present, and on inner aspect you can see the pharyngeal pouch which is present. In this diagram you can see this is the area of the stomodium, and in this blue color you can see the oropharyngeal membrane. So you can see this one is the mandibular prominence and the maxillary prominence. These both prominences are part of the first pharyngeal arch. Again, in further development you can see this one is the maxillary prominence and then the mandibular prominence, and here you can see oropharyngeal membrane now rupture, and there is an open communication of the oral cavity with the amniotic cavity now. And here in this diagram with further development you can see the nasal placoids have developed, the first pharyngeal arch, the second, third and fourth are present, and this one is the opening of the oral cavity, the stomodium which is present. So what is the fate of the pharyngeal arch? This is very important. The pharyngeal arch contribute exclusively to formation of the face, the nasal cavity, the mouth, the larynx, pharynx and neck. So during 5th week, the second pharyngeal arch enlarges and overgrows the third and fourth, forming the ectodermal depression called the cervical sinus. So in this diagram you can see this one was the first pharyngeal arch and this one is the second pharyngeal arch. This second pharyngeal arch grows downwards and it covers the third and fourth pharyngeal arch, and this area, this sinus which is entrapped behind the second pharyngeal arch is called the cervical sinus. By end of the second week, second to fourth pharyngeal grooves and cervical sinus disappear, giving the neck a smooth contour. So this is the fate of the pharyngeal arches. Now, what components which are forming the pharyngeal arch? So, its mesenchyme develop basically into 4 components. Its own cartilage. So, every pharyngeal arch has its own cartilage, its own muscle, its own nerve and artery. So, this is also very important. Whenever some muscle move, they migrate from the development of the pharyngeal arch. They carry their nerve supply along with them, and because of this region they can be traced. Their origin can be traced, that they belong to which pharyngeal arch. So now we will discuss the first pharyngeal arch. This first pharyngeal arch has 2 components. In purple color you can see this is the maxillary prominence and this one is the mandibular prominence. So this first pair of the pharyngeal arch plays a major role in the facial development. What structures are forming? First you have a look at this diagram. You can see that these are the areas of developing arches and pouches, and here you can see these are the aortic arches which are forming, and each aortic arch gives branch to its related pharyngeal arches. So first to fourth aortic arches giving branches to first to fourth pharyngeal arches. Now, first pharyngeal arch has maxillary process and the mandibular process, and the maxillary process forming the premaxilla. Here, this area, in this area, the premaxilla is forming a small piece of the bone, which later on fuses with the maxilla, and from here the incisive tooth is attached. Then the premaxilla, then the maxilla which is formed from is, and the zygomatic bone and part of the temporal bone. So these are formed from the maxillary process. These are the skeletal components of the first pharyngeal arch. The mandibular process is also very important. It has dorsal end and the ventral end. So dorsal end of the mandibular process ossifies and it forms the malleus and the incus. The middle part of this cartilage becomes the anterior ligament of the malleus and then the sphenomandibular ligament, and the ventral part of this cartilage, it will form the Meckel's cartilage. Later on, around this Meckel's cartilage the mandible will form. So the skeletal component, again I repeat, The mandibular, the maxillary division, the maxillary process from the premaxilla, the maxilla, the zygomatic bone, and part of the temporal bone. And the mandibular process from the incus, the malleus, the anterior ligament of the malleus, the sphenomandibular ligament, and the mandible. So these are the skeletal components which are derived from the first pharyngeal arch. Now we move on to the second pharyngeal arch. The second pharyngeal arch, its cartilage is called the Riker's cartilage. It ossifies to form the stapes and then the styloid process of the temporal bone. These two processes are formed from the second pharyngeal arch, then the stylohyoid ligament which is formed, then the lesser carnu and the superior part of the body of the hyoid bone is formed by the second pharyngeal arch. So these are the components. Then the pericardium which is forming the stylohyoid ligament. So these are the components from the second pharyngeal arch. The stapes, the styloid process of the temporal bone, the stylohyoid ligament, and the lesser carnu and part of the superior part of the body of the hyoid bone, which are forming from the second pharyngeal arch. Now the third pharyngeal arch, this third pharyngeal arch, its cartilage ossifies and it forms the greater cornu of the hyoid bone and most of the part of the body of the hyoid bone. So these are the derivatives of the third pharyngeal arch. Then the fourth and the sixth pharyngeal arches, their cartilages fuses to form the laryngeal cartilages except the epiglottis which develop from the hypopharyngeal eminence. This is very important from MCQ point of view because you have to memorize all of the laryngeal cartilages are formed from fourth and sixth except the epiglottis which is formed from the hypopharyngeal eminence. Fifth arch is usually rudimentary and it has no derivatives. So here you can see the derivatives of fourth and sixth pharyngeal arches forming the thyroid, the cricoid, the arytenoid, the corniculate, the cuneiform cartilages. Over here the different cartilages of the larynx except that of the epiglottis. So now we move on to the muscular components which are formed from the different pharyngeal arches. The first pharyngeal arch forms the muscles of the mastication. Then it is forming the anterior belly of the digastric muscles. Then it is forming the mylohyoid muscle over here. Mylohyoid muscle, and then it is forming the tensor tympani and tensor palatini and part of the temporalis muscle. So these are the temporalis muscle, the masseter muscles, the mylohyoid, the anterior belly of the digastric. These are formed by the first pharyngeal arch. The muscles are formed by the first pharyngeal arch. The second pharyngeal arch usually form muscles of the facial expression. So in dark pink color you can see the orbicularis oculi, the frontalis muscle, the orbicularis oxyli, the orbicularis oculi, the buccinator muscle, the orbicularis oris muscle, and also the part of the auricularis muscle which is present and occipitalis muscle. So these are formed from the second pharyngeal arch, and also very important is the posterior belly of the digastric muscle. Remember please, anterior belly of the digastric is formed by the first pharyngeal arch and posterior belly of the digastric muscle is formed from the second pharyngeal arch. The muscle which is formed from the third pharyngeal arch is only the one muscle. Here it is shown in blue color. This is the stylopharyngeus, the only one derivative of the third pharyngeal pouch. The fourth pharyngeal pouch forms the cricothyroid, the levator villi palatini, and constrictors of the pharynx. The sixth pharyngeal arch form intrinsic muscles of larynx. You have to memorize that all the intrinsic muscles of the larynx are derived from the sixth pharyngeal arch except the cricothyroid, which is derived from the fourth pharyngeal. So this is also the point of difference of the nerve origin as well, because cricothyroid has the nerve supply of the fourth pharyngeal arch, and all other muscles, the intrinsic muscles of the larynx, have the nerve supply from the sixth pharyngeal arch. So now the derivatives of the pharyngeal arch nerves. The first pharyngeal arch has two parts, the maxillary and mandibular, and the maxillary and mandibular parts of the first arch is supplied by the maxillary and mandibular branches of the trigeminal nerve. The second arch is supplied by the facial nerve. The third arch is supplied by the glossopharyngeal nerve, and fourth and sixth are supplied by the branches of the vagus nerve. So the branch of the vagus nerve which is supplying the fourth arch is superior laryngeal branch, and this is the reason why the cricothyroid muscle is supplied by the superior laryngeal artery of nerve of the vagus nerve. The sixth nerve is supplied by the recurrent laryngeal branch, and this is the reason all intrinsic muscles of the larynx, they are supplied by the recurrent laryngeal nerves because they are derived from the sixth arch, except that of the cricothyroid muscle, which is supplied by the superior laryngeal branch because it is derivative of the fourth arch. So now here you can see the different arches and their nerves. The first arch, both the maxillary and the mandibular process, which are supplied by the maxillary and mandibular division of the trigeminal nerve. The second arch, which is supplied by the facial nerve. The third arch, which is supplied by the glossopharyngeal nerve, and fourth and sixth arches, which are supplied by the branches of the vagus nerve. So now we move on to the pharyngeal pouches and their derivatives. The primordial pharynx derived from the foregut widens cranially where it joins the primordial mouth or the stomodium. It narrows caudally where it joins the esophagus. So endoderm of the pharynx lines the internal aspect of the pharyngeal arches and passes into balloon-like diverticula called the pharyngeal pouches. So now we move on to pharyngeal pouches and clefts. Here in this slide you can see the four pairs of the pharyngeal pouches are formed. The first pair is formed between pharyngeal arch first and second, and the second pair pouch is formed between second and third arch, and third pouch is formed between third and fourth, and similarly the fourth pharyngeal pouch. These pouches are lined by the endoderm of the pharynx which is present over here, and they are forming the different derivatives. So the first pharyngeal pouch will form the tubotympanic recess. Its distal part will form the middle ear cavity, and its proximal part will form the eustachian tube. The second pharyngeal pouch, which is present over here, it will here the palatine tonsils will form. Its epithelium infiltrate the local area of the mesenchyme and form the palatine tonsils. Later on, the lymphoid tissue infiltrate this area. So this over here the palatine tonsils will be formed, and the original pouch which was present is now shown as the fossa for the palatine tonsils. Now we move on to the third pharyngeal pouch. This third pharyngeal pouch has two parts, the dorsal part and the ventral part. The dorsal part forms the inferior parathyroid gland, and the ventral pouch part of the third pouch will form the thymus gland. So with the development of the thymus grows distally and downwards to come to its adult position in front of the neck and the chest. So it also brings the inferior parathyroid gland along with itself, so that it comes to lie behind developing thyroid gland. The fourth pharyngeal pouch will form also has the two components, the dorsal and the ventral component. Dorsal component will form the superior parathyroid, and the ventral component will form the ultimobrachial body or ultimopharyngeal body. So this superior parathyroid gland also moves along with the developing thyroid gland, and it comes to lie dorsal to the upper part of the thyroid gland. So in that is why this is the region that the superior parathyroid is derived from fourth and inferior parathyroid is derived from the third. It is given the name inferior because it lies inferior to the superior parathyroid gland, but it is derived from third pharyngeal pouch, and superior is derived from the fourth pharyngeal pouch. The fifth pair is absent or rudimentary. Now we move on to the cleft which are present on outer side, or the grooves which are present on the outer side. So this one is the first groove, the second, third and the fourth groove. So please dear student, remember one point, that in previous books it was mentioned that the first pharyngeal groove will form the external auditory meatus. Now this concept has been challenged And it is now evident that actually the ectoderm of the first pharyngeal arch, ectoderm, remember, ectoderm of first pharyngeal arch invaginates and it forms the external auditory meatus. It is not the pharyngeal cleft. So please, this is a change in the concept which is written in the new books. You can see in the newer editions of the books that this pharyngeal groove, first pharyngeal groove or cleft, is formed by the ectoderm of the first pharyngeal arch. The second, third and fourth pharyngeal cleft, here you can see the second pharyngeal arch overgrows and it comes to lie downward and it covers the cleft of the second, third and fourth cleft, and it covers and completely, when it completely covers these cleft, the neck will have a smooth appearance. So the cleft which were present over here, later on they will form the cervical sinus, and the cervical sinus later on disappear. So the only here you can see only the external auditory meatus is present, and this external auditory meatus is derived from the ectoderm of the first pharyngeal arch. So these are the different derivatives of pharyngeal arches, pouches and clefts over here. So here in this diagram you can see this is the first pharyngeal pouch forming the pharyngotympanic tube. Its distal part will forming the middle ear cavity and the eustachian tube. The second pharyngeal pouch which is forming the tonsillar fossa over here. The third pharyngeal pouch forming the inferior parathyroid and thymus gland, and fourth pharyngeal pouch which is forming the superior parathyroid and ultimobrachial body. Here you can see the area of the developing tongue. You can see over here, and these are the derivatives of the pouches. Now we move on to the pharyngeal membranes. Pharyngeal membranes appear in floor of the pharyngeal groove. These membranes form where the epithelium of the groove and the pouches approach each other. The endoderm of the pouches and the endoderm of the grooves are soon separated by mesenchyme. Only the first pharyngeal membrane becomes the tympanic membrane and other obliterate. This is again your MCQ. So please memorize that only derivative of the pharyngeal membrane is the derivative of the first pharyngeal membrane, which is forming the tympanic membrane. This tympanic membrane lines on the outer aspect by the ectoderm, on the inner aspect by the endoderm, and in between these two there is the mesenchyme which is present. So now we move on to the congenital anomalies, the remnants of these pouches, arches and sinuses. So first you can see this was the first pharyngeal groove and first pharyngeal arch, second pharyngeal arch, third, fourth and sixth arches. So second pharyngeal arch grows downward and overgrows, and it covers the third, fourth and sixth, and as a result it also covered the developing cleft, the third, fourth and the sixth cleft. So if a connection, when this cervical sinus is formed, later on this cervical sinus also obliterates. So if this cervical sinus persists, here you can see this is the tract of the cervical sinus into the area of the tonsils, into the area of the pharynx. So if this cervical sinus persists in the cranial part, it will form the internal brachial fistula and also the internal brachial sinus in the area of the pharynx. And if it persists on the outer aspect, it has a connection with outer area, it will form the external brachial sinus. So here this external brachial sinus, if it has a communication with the internal brachial sinus, this will call the fistula. And here you can see the most commonest location of the fistula is the just anterior to the sternocleidomastoid, behind the angle of the mandible. So these are the openings. The other locations are the openings of the brachial vestiges, or sometimes the brachial sinuses which are present over here. So here you can see the triangular area where the auricular pits may be present. So these are the different remnants of the cervical sinus which are related to the development of the pharyngeal clefts. Here I complete my today's class, and the references are Langman Medical Embryology, The Developing Human by the Keith Elmore, and The Gross Anatomy by the Susan Stranding 40th edition. Thank you very much for your attention, and we will meet in the next class with the development of the face and tongue. Till then, Allah Hafiz.